transcription unit and gene. Before we learn the transcription unit, we must know what is a gene. Gene is a part of DNA. A small part of DNA carrying information for the production of one protein. This is called as gene. So this carries information for the formation of one protein, and this is followed by another gene. Now you can understand what is gene. Let us see what is transcription unit. This is a structural gene. The structural gene has two strands. The two strands are opened by the breakage of hydrogen bonds. Of the two strands of DNA, only one strand is copied into mRNA. That is called a transcription. The process of copying genetic information from one strand of DNA into RNA is termed transcription. Both the strands are not copied into RNA. After mRNA formation inside the nucleus, the mRNA moves through the nuclear pore and reaches cytoplasm. Based on the information on mRNA, the ribosomes produce proteins. This is a structural gene. This is beginning of the structural gene. That is the beginning of transcription. Transcription begins at the beginning of structural gene. This is the end of the structural gene where transcription ends. Structural gene has the polarity of 3' and 5'. The DNA sequence before the structural gene is promoter, and the DNA sequence after the structural gene is terminator. Transcriptional unit in DNA is defined by three regions: a promoter, a structural gene, and terminator. The two strands of the structural gene have opposite polarity, 3', 5', and 5', 3', but they are complementary to each other. They are complementary nucleotides: adenine, thymine, thymine, adenine, guanine, cytosine. The first one is template strand. The polarity of template strand is 3', 5'. The template strand will be copied into mRNA. Another strand is coding strand. Coding strand at the ends contain promoter and terminator. And the newly formed mRNA is complementary to the template strand. Now see, this is newly formed mRNA. It is complementary to the template strand. Thymine, adenine, guanine, cytosine. But here adenine, thymine will not come. Thymine is replaced by uracil. And the base sequence of coding strand and mRNA are similar, with the exception of thymine replacement by uracil. See, other things are similar. And both the strands, both the polynucleotides, that is coding DNA and mRNA transcript, are complementary to template strand. T is in template strand. Complementary nucleotides of T, adenine. Adenine is in coding strand as well as mRNA. So these two are coding strand and mRNA are complementary to template strand. What's the location of promoter? This is a location of promoter. That is 5' end of coding strand. Promoter is located at the 5' end of coding strand. Terminator is located at the 3' end of coding strand. And what's the role of promoter? The promoter is a nucleotide sequence that is found before the structural gene. It provides place for the attachment of RNA polymerase. Okay, what's the role of terminator? The RNA polymerase starts reading the structural gene from this area. When it reaches the terminator, it stops reading. The terminator stops the transcription process. That is the end of transcription. In eukaryotes, the promoter has AT-rich regions, that is thymine and adenine-rich regions, called Tata box or Goldberg-Hockness box. AT-rich regions are called as Tata box. In prokaryote, the same region is called Pribnov box. In eukaryotes, in addition to the promoter, there is a need for enhancer. Enhancer also involves in transcription in addition to promoter in eukaryotes. The Tata box in eukaryote is called as Pribnov box in prokaryotes. Now we have learned the